Okay guys, welcome back. Today we are going to be going over quite a bit. Okay, so to start off, you guys are doing really good in the class so far. I know taking these online exams can be somewhat difficult. I'm going to try and make them a little easier. I know the step-by-step process of delineating out that one red blood cell following through the heart. It can be a little difficult as far as what everyone was choosing. So moving forward, I'm hoping that we can dial in this test taking and find a middle ground as far as assessing your guys' knowledge as well as the test being fair for everyone. So that being said, I'm going to try and play with the tests here and make them a little bit more user-friendly moving forward. Today, we are going to go over heart electrical conductivity, the physiology of the heart, how electrical conductivity works, and then we're going to go over an ECG or an EKG wave, electrocardiogram wave. We're going to break down the wave. I'm not going to do the timing of the wave. I'm not really going to go over the timing of this, although it happens very fast. I will briefly put up a couple of slides and pictures that talk about timing so you can see how fast it is, but I'm not going to assess you on that knowledge, so that's not going to be on the quiz. However, how the step-by-step process of electrical conductivity will be on the quiz. Also, labeling the P wave and labeling all the segments will be on the quiz. Understanding what the heart anatomy is doing during the ECG wave will also be on the quiz. And then lastly, we will finish up with blood pressure and talk about blood pressure, a basic overview of blood pressure and what exactly blood pressure is and how to assess blood pressure. Another thing I forgot to mention before we move forward into this is the resources. So on this week's module, we have a good amount of resources available. I want to make sure that you guys are going through all those resources. So there's several packets of information and several videos. I found these videos online. They're really good videos, much more high-tech than mine, and they go into detail with explanation. So utilize those videos. What I want you to do is open up your lab assignments. There's 2 lab assignments this week. Heart conductivity, ECG. Then there's also the blood pressure assignment. Okay, a lot of that will be taken out. You'll see that. There's also a blood pressure information packet with study questions. Do those study questions, all right? Use all your resources to be able to answer those study questions. Hey, Google's a resource. Let's Google it. Your book is a resource. Yes, you have a book. We can open it. That's good. So use all the resources that you have available to you, okay? Study. Knock out your lab assignments. Then at the finale of this week, take your lab quiz. Don't take the lab quiz and then do the assignments because then that defeats the purpose, okay? Practice the knowledge, practice the material, then we assess you at the end, all right? All right, let's go ahead and get started now that I made that little public service announcement with the lecture. The heart, my lovely rudimentary drawing. You need to do this. This is part of your assignment. We're going to draw this again. You'll notice, got my handy dandy tool. We have right atrium, left atrium, right ventricle, left ventricle. So, how does the heart contract? How does electricity travel through the heart? Besides this lovely drawing that I have here for you that you also need to do as part of your lab assignment this week, I'm going to be going over some slides that I found on the internet that are open slides, so hopefully I can use them on this video. I'm going to use them anyways to help better illustrate how this occurs, because sometimes this can be confusing. This is all about flow of ions, negative and positive, and depolarization and repolarization. Okay, so right off the bat, let's talk about this. First of all, the heart works off of an electrical system. Okay, depolarization and repolarization. If you remember way back when to early biology classes, we learned about depolarization and repolarization. Okay, depolarization is basically when we have an influx of positive ions into the cell. And then that depolarization occurs, which causes muscle excitation. Remember, contraction occurs. Atrium's first, ventricles next. Lub, dub. So how does that occur? Excuse me. It all starts with that guy right there, the SA node. The SA node, this guy, stands for sinoatrial node. Okay, so let me go ahead and write that up here. All right, so here, this guy right here is our sinoatrial node. It is our SA node, sinoatrial node. This node is a clump or grouping of cells found in the right atrium. This clump or grouping of cells is nervous tissue. Okay, the role of this is the internal pacemaker. It's the first pacemaker of the heart. It's what sets the stage for the heart to have good solid contraction and timed very well. Okay, so when we have the SA node fire off, so the threshold is met, action potential occur. So sodium is flowing into the cells of the SA node. It reaches an action potential that opens up the calcium channels, which is also another positive ion. That then causes an exponential rise in excitation or electrical conductivity. And those calcium channels open up. Calcium flows in, causes depolarization to occur. That depolarization occurs, reaches over a certain threshold, and then potassium channels open up. Potassium then flows back in to the cell, causing sodium to flow back out. Sodium, potassium, okay, shifts across the cell membrane. That causes the action potential occur. Here's where timing is really important, and then we'll go over a slide with timing as far as how it occurs, and I might hop back and forth between that slide and have an overlay of voice so that we can kind of explain it further in detail. Okay, let's just talk real quick. I decided just to go ahead and do this overlay. This is a photo that I got from the interweb. All right, so what's going on here? Timing of the heart. So remember, the SA node, the sinoatrial node, depolarizes, and a depolarization wave effect occurs down through Bachmann's bundle and the internodal tracts, and those go through and cause the contraction or result in the contraction of the atriums and then down to the AV node. So if we look at this diagram here, we can see the timing of it all. Very fast this occurs, right? Roughly 60 beats a minute is what our heart does. Lub dub, lub dub, lub dub. And if we notice here the SA node, we have timing. Well, approximately 0.09 seconds it takes for the electrical conductivity or the depolarization to reach and maximize in the left atrium, and approximately 0.07 seconds for the electrical conductivity to reach the AV node. Okay? From there, it continues on. Before I get ahead of myself, because we have to go back to the diagram of what we were drawing, let's end it there. So there you see a little brief overview of timing of it all, 0.07 seconds to 0.09 seconds. Atriums are depolarized and contracting. But for the here and now, let's talk about this, okay? So SA node depolarizes. When the SA node depolarizes, we have electrical conductivity moving through. Moves really fast through certain nervous tissue fibers. We have a nervous tissue fiber that comes across the right atrium through into the left atrium, and this bundle of nerve fibers is called Bachmann's bundle. Bachmann's bundle, okay? That causes the electrical conductivity to flow. Electrical conductivity also flows within the muscle cells, okay? However, it is not as fast as it flowing through the Bachmann's bundle, through nervous tissue, all right? It's a little bit slower, okay? Another thing that is important to know is the nervous tissue that flows down. We have three bundles, three tracts of nervous tissue that come from the SA node and come downwards, okay, towards our next pacemaker, okay? Our next pacemaker is right here. These three bundles or three tracts that come downward towards that pacemaker along the right atrium, those three tracts are called the Internodal Tract. I know that's hard to read and you all love my handwriting, okay? But this is called the Internodal Tract. Nodes, SA node, here, AV node, okay? The AV stands for atrial ventricular node. Okay, I'm going to say that again. Atrial ventricular node. Atrial meaning atriums, ventricular meaning ventricles. Pretty easy to remember, right? So internodal tracts, those are the three tracts that connect or transmit electrical conductivity to the AV node and also helps with depolarization. Get my handy dandy tool out again. So from here we have the SA node releases or depolarizes. All those ions flow into the cell causing depolarization. This travels across Bachmann's bundle causing contraction of the left atrium pretty close to the same exact time that it occurs in the right atrium, okay? And then electrical conductivity from the SA node travels down the internodal tracts, also causing exponential contraction of the right atrium or electrical conductivity to travel through the right atrium. Then it also travels to the AV node. The AV node then transmits electrical conductivity from the atrioventricular node to a very, very important set of tissue, okay? This set of tissue right here, you notice that it's a little bit larger and highlighted color. This set of nervous tissue is called the bundle. Let me put a line here separating these two. But this set of tissue is called the bundle of His. The bundle of His. Okay? This bundle of His has a very important role. Okay? The short of the long of it is the bundle of His slows down the electrical conductivity. And how does it slow it down? Well, it has to do with it has a low permeability rate of sodium. So sodium rushes into the cell, okay, or flows into the cell that then results in an action potential for the calcium channels to open up so that calcium floods into the cell and causes an action potential to occur, okay, depolarization. Excuse me. What the bundle of His is, because the permeability of sodium is lower, sodium doesn't flow in as readily into the bundle of His. So the action potential takes a little bit more time to occur. Since the action potential or the depolarization takes a little bit more time to occur, this slows down the timing of the contraction, which is good. We want that. We don't want our atriums and our ventricles to contract at the same time because then all of a sudden we're getting boom, contraction, contraction, right? That's not good. So instead what we want is atriums to contract. This slows down right through here. Okay, so the bundle of His slows that down. And then we reach these two branches, our right and our left branch, for electrical conductivity to occur. This center part right here, okay, this interventricular septum, all right, this is called a septum. This is where the two branches, right and left branch, use my pointer tool so you can see everything. The two branches, the right and the left branch, then speeds back up electrical conductivity. Why? Because sodium permeability is a little bit higher through these fibers, okay, than the bundle of His, all right? So the electrical conductivity flows down the interventricular septum, or the right and left branches, comes all the way down, and then splits, okay? Splits so it can go to the right and the left ventricle relatively even, okay, as far as timing is concerned. What we don't want, and you'll notice that they come down and around and then release, all right? And the reason why is because we want our ventricles to contract upwards, right? Because we have our pulmonary trunk and our aortic trunk right there. We need blood to go up into the lungs, and then blood to go up into the aorta to then go back down and go throughout the entire body. All right? So we don't want these two fibers to come out and, pointer tool, go to our right and left ventricle here. Okay? We want it to travel down the septum and come around and then enter the right and left ventricles here. Now, really important. Remember, if I write something on the board, it's really important, even though my handwriting is terrible. All right? As the electrical conductivity goes down the right and left branches, we meet these fibers that innervate the right and left ventricle. Okay? These fibers are called, important here, asterisks, Purkinje fibers. Purkinje fibers. Okay, these fibers are called Purkinje fibers. All right? So, that is a P. Purkinje fibers. Purkinje. I misspelled it the first time, so I had to redo it. Okay? All right. Purkinje fibers. These Purkinje fibers release the electrical conductivity, or the depolarization, in the ventricles, in the tissue, the muscle tissue of the ventricles down here, and then contraction occurs. All right? So, how does contraction occur? Well, let me get my marker again. SA node sets the pace of the heart. It's the internal pacemaker, along with the AV node. It's another pacemaker of the heart, and the bundle of His is considered a pacemaker of the heart as well. We have depolarization, goes across the Bachmann's bundle, comes down through the internodal tracts, causes contraction of the ventricles. So, all these negative guys here, we get positive into the cell. Boom, boom, boom. We're going positive. We're going positive. Okay? Then, as that electrical conductivity comes down the internodal tracts, reaches the AV node. The AV node then depolarizes into the bundle of His. The bundle of His slows down conductivity. That was not slow motion, that was me. The bundle of His slows down conductivity due to sodium permeability being low. Then that electrical depolarization goes down the right and left branches, out to the Purkinje fibers, and then, boom, ventricles contract, and we flow upwards, right? We want blood to flow out the aortic trunk and the pulmonary trunk. This right here is heart electroconductivity physiology 101. All right. Now let's look at some diagrams and let's talk about those. And then what I'd like to do is draw out the ECG wave and talk about the ECG wave and talk about the physiology of electrical conductivity as well as the anatomy of the heart and what's going on in the heart during the ECG wave. Side note, once depolarization occurs, we have to what? I asked you a question and I'm in a room all by myself, so that's weird. But we have to repolarize, right? Repolarization, the sodium-potassium pump. Think about those dominoes set up. You flick that first domino, they all start to fall down, and then somebody comes back up behind the dominoes and picks them back up. That's the way I like to think about depolarization. Pretty easy, simplistic way to think about it. That sodium-potassium pump is what then shoots sodium, potassium back through that helps pick back all those dominoes back up so that we can depolarize again, okay? So repolarization is when the ions flow back out of the cell, so sodium flows back out of the cell so that we can repolarize so that we can contract again. Interestingly enough, when atriums contract, depolarization occurs, comes down, then ventricles contract. During this contraction, the atriums are actually repolarizing, okay? And we'll talk about that in the ECG wave. All right, here's a diagram I found on the interweb, and it was a physician who did a talk on the electrical conductivity and how depolarization occurs. And this specifically I really like. This was a really cool diagram. They kind of gave an example of the sodium permeability, the threshold, and basically the length of time that it takes for the action potential to occur with each pacemaker. Okay, so let me explain this diagram. First, on the left-hand side, on the y-axis, we have millivolts, all right? And then on the x-axis, we have time. Okay, so the amount of time that it would normally take. The yellow line denotes the SA node, sinoatrial node. Sodium permeability is relatively good there. So as sodium enters the cell, millivolts start to increase within the cell. It finally reaches the threshold, that pink line there. Once it reaches that threshold, the calcium channels are open, and then we get a really fast increase or depolarization then occurs, and then that's that contraction of the atriums, okay? Once it passes a certain line or a certain amount of millivolts, then potassium channels open up, helping to restart the process, and then repolarization occurs. Next, we can see that the sodium permeability is a little bit more, or excuse me, a little bit less for the AV node. So the AV node takes a little bit longer. So when the depolarization reaches the AV node, it takes a little bit longer for the AV node to finally reach the threshold for an action potential to occur, depolarization. And then we have that spike or increase. That's that green line going up once it reaches the threshold there. And then we have depolarization of the ventricles, aka ventricular contraction. Then the potassium channels open back up after a certain limit. And then we have repolarization. Lastly, there we see the bundle of His, denoted as BO or little o, H. And then the bundle of His, the sodium permeability is very, very, very low, which causes, which is good because the timing then slows down. We don't get contraction of the ventricles immediately after the AV node depolarizes, so it slows down, and then we finally get those calcium channels opened up once we reach past a certain point of that threshold, and then depolarization occurs, causing that electrical conductivity to go down the interventricular septum over to the Purkinje fibers so that we can get a strong ventricular contraction. Okay, before we go into the actual wave of the ECG, let's do that step-by-step process because you may have a question regarding the steps. All right, so I'm going to just go ahead and write the steps on the board, and we'll see if maybe we can go through it really fast once I write the steps on the board, all right? All right. Step 1, SA node depolarizes, okay? Contractions are going to occur. Well, how? Depolarization of Bachmann's bundle goes over to the left atrium. Internodal Tracts helps to depolarize the right atrium and also transmits electrical conductivity really quickly to the AV node. Step 3, atrial depolarization occurs. Step 4, AV node depolarization occurs. Step 5, bundle of His depolarizes, slows down conductivity, okay? Step 6, depolarization of the right and the left branches, goes down the interventricular septum. Step 7, Purkinje fibers depolarize. Step 8, depolarization of the ventricles. Not a step 9, but a happy heart afterwards, okay? That is the step-by-step process that you may or may not have a quiz question on. You're probably going to have one. Here is our ECG or EKG. There really is no difference between ECG and EKG. The one difference is the K is Jamin, okay, and this C is what we call ECG, electrocardiogram. It is the electrical conductivity of the heart. It's the way we measure the electrical conductivity of the heart and help to determine what is going on with the heart, okay? So if we see an issue with this wave, or this wave, or this wave, or the timing, or the height, or the distance, or the closeness of it, we know there are certain things going on with the heart because this is directly correlated to the anatomy of the heart doing something, the physiology, okay? So first, let's label our waves. We have the P wave, we have the Q, the R, and the S wave. We call that together QRS complex. And then we have the T wave. By the way, this is one of your lab assignments, so you're going to have to do all this, all right? So let's name everything first, like we're doing, and then let's talk about what's going on in the heart while these are going on. So the P wave, Q, R, S complex, and the T wave. Well, we also have some segments and some intervals, okay? So from our P wave to our Q wave, we have the PQ interval, okay? We can either measure from the start of the P wave or we can measure from the end of the P wave. It's going to tell us different things. The height and the magnitude of the P wave has to do with atrial depolarization, okay? So the PQ interval. Then we have our QRS complex. From here, the S to the T. So the end of the S wave to the beginning of the T wave is known as the S and T segment. Okay, the S and T segment. Okay, another thing. PQ interval is also known as the PR interval. That's how we really call it, is PR interval, but for students, usually it's easy to remember PQ interval. So PR interval. This section right here, if we go from the end of the P wave to the beginning of the Q wave, we actually call this the PR segments. Okay? S and T segment is here. And we can also take this segment and go to the end of the T wave, and this is actually known as the S and T interval. Okay? We can do from the Q wave down past the T, QT interval. Okay? There's many, many, many segments and intervals that we can label and study and look at. And it's all about the magnitude and the timing. Okay, magnitude of the waves, timing of the waves. All right. So my PQ interval, if this starts to lengthen, well, that's a problem. If my S&T segment starts to lengthen, that's a problem as well. All right. So first and foremost, the P wave. What is the P wave? Well, the P wave is atrial depolarization. The QRS, you notice that the magnitude is quite large. Well, that is ventricle depolarization. And the T wave is ventricle repolarization. Okay? So, we have atrial depolarization, ventricle depolarization, and ventricle repolarization. Okay? The PR segments, all right, the time after atrial depolarization to the beginning of ventricle depolarization, that is the time that it takes for this to occur. There's a specific timing. If this is shorter, then that means we have something going on with the nodes of the heart and the electrical conductivity of the heart, that electrical conductivity is occurring too fast. And that's not good, because that means that we're going to have not enough blood ejecting into the aortic trunk and the pulmonary trunk, okay? Because there's not enough ejection time in between atrials contracting and ventricles contracting. Remember, when we talk about depolarization, that's just another way of saying contraction. So what's occurring in the heart is the atriums are contracting, QRS, ventricles are contracting, okay? Time in between that from occurring is right here. Time in between ventricles contracting and ventricles relaxing and repolarization, maximum repolarization, is right here, the S&T segment, okay? One thing that we don't see is atrial's repolarization, and that's because it's hidden behind the QRS, okay? When ventricles are depolarizing or contracting, at that same time, the atriums are relaxing and repolarizing, okay? This is a very important concept, because we want our heart to do what? Continue to beat, right? So we have lub-dub, lub-dub. Well, that lub-dub that occurs when the dub, the QRS, contracts, we need the lub to be able to relax, okay? So, ECG, all right, this is what we're looking for. And there's a bunch of timing. So let's look at some of those diagrams. Okay, let's go over now this diagram. This is a pretty good one, a lot better than my drawing. Here's a really good explanation on the P wave, the PR interval, QRS complex, the S&T segment, and the T wave. Okay, so we can see depolarization of the atriums at the P wave. The QRS complex is depolarization of ventricles. The main pumping contraction of the ventricles helps to eject blood out of the left and right ventricles into the aortic trunk and the pulmonary trunk, right? Really important stuff there. We don't get to see the atrial repolarization. Remember, that occurs behind the QRS complex. The QRS complex is so large in magnitude that we really can't see atrial repolarization, okay? The S&T segment begins ventricle repolarization, and maximum ventricle repolarization occurs at the T wave, okay? All right, so that's a pretty good image right there. The next image that I'm just going to show you is just going to be timing. Once again, remember, I'm not going to test you on timing, but you'll be able to see two ECG waves lined up right next to each other, and the different intervals and segments, and then you'll see the timing of it all. So I'll leave that slide up there for a little bit, but that concludes electrocardiogram. Okay, let's talk a little bit real quick about blood pressure. Okay, I like to describe blood pressure as an example of, well, a pump station, right? The heart, the cardiovascular system, it is a closed system. So the way that blood flows through our entire body is not by the heart pumping it through the whole body. That's a common misconception. The heart is the main pump house. It maintains pressure in the system. For instance, our city, we have waterworks, and our city uh, provides water for the city. Well, we don't have a pump in our house that's pumping water through our house. Water gets to our house and goes to all the pipes in our house because there is pressure within the system. There's pressure maintained within the system. So the heart and our body's the same way. It's a closed system, and there's pressure maintained within the system. That pressure that's maintained in the system is done by three things. Number one, it's done by the main pump. The main pump is our heart, right? So that's the main pump. Maintaining pressure in the system, really maintaining the bulk of the pressure in the system. Two other ways that pressure is maintained in the system is these blood vessels. Okay, this is just a rudimentary drawing or a schematic, if you will. These blood vessels have muscles within them. Arteries have muscles within them, okay? And so the muscles within the arteries help push or flow or movement of blood throughout the entire system, okay? So that's why we have pulses, is because our arteries are actually contracting and helping blood move throughout the entire system, okay? So this right here kind of contracts inwards, all right? And so these are surrounded by smooth muscle that helps to contract and pulse blood through, all right? The third way that pressure is helped maintain in the system is called muscular systole, and we'll go over all the systoles and diastoles and what that means here in just a second, but I want you to understand these concepts first. So our muscles help to move blood or maintain pressure in the system, specifically in our veins, okay? Veins are strategically placed around our body just naturally as we grow. Our veins are strategically grown throughout our body in between large gross muscles, okay? And what those muscles help to do is those muscles help to create pressure within our veins, all right? As we move, our muscles contract, expand and contract, and then that helps push on our veins to help blood flow through. So pressure is maintained within the closed system, the cardiovascular system, by the heart, the muscles in our arteries, and our skeletal muscles to help push blood throughout our cardiovascular system. Okay, let's talk about that smooth muscle and that skeletal muscle and how arteries and veins help to maintain pressure in the system. Okay, so like I said, arteries, within the diameter of the artery, we have smooth muscle, okay? And this smooth muscle helps to contract, okay? Helps to contract and push the blood, or doesn't help to contract, it contracts and helps to move blood throughout the cardiovascular system, okay? So we have our nice little red blood cell flowing through the artery, and then we have contraction occurring, okay? That's why we have a pulse. And we'll talk about the different pulses that we have in our body. And in your lab, I actually want you to find specific pulses within your body and then see what the beats per minute is as compared to the other pulses in your body, okay? Because as we get farther away from the central part of our heart, our pulse is going to be a little bit different as far as the timing of when you feel the beats as compared to when it's going on in your heart. Okay? Okay, so veins, the way veins help to maintain pressure in the system are twofold. One, the venous valve, and then two, skeletal muscle. So as we move and walk around and do things, skeletal muscle contracts and presses up against the venous valve and helps blood flow. Well, these valves, the way that they work is they help to prevent backflow. Really important. Okay, so venous valves, the job of venous valves is to prevent backflow. Okay, help prevent backflow. So this is what we don't want. We don't want this red blood cell going back down once that valve is closed. So the valve opens, red blood cells flow up, valve closes, we can't go down. We don't want backflow to occur. If we get damage to this venous valve, then pressure increases in this area. If too much pressure increases, then this valve hardens and gets damaged, and then we have more backflow, and then more backflow, and then more backflow. And then eventually we have circulatory problems and problems within our veins. Then our veins start to harden, and they get a little squiggly, okay, by view. And then we have basically too much blood pooling within that vein that can then lead to a condition called varicose veins. Okay, ladies, one of the ways that we try and avoid varicose veins is don't sit with your legs crossed. If we sit with our legs crossed, you'll notice that you lift up one leg and you have a giant red mark on maybe your calf. Okay, that giant red mark in your calf is you occluding those veins. When you occlude those veins for too long of a time, you build up backflow pressure. That backflow pressure can damage these venous valves and then long term can result in varicose veins. So note to self, sit with your legs together, not crossed. If they're crossed, you can actually cause damage to your venous valves by increasing the amount of pressure in your veins. Okay? So, recap, arteries have smooth muscle around them. That helps blood flow throughout the cardiovascular system. Those are our pulses, okay? Venous valves helps blood flow, or actually to prevent backflow, so thereby helping blood flow in the right direction, okay, towards our heart. All right? Skeletal muscle helps push and maintain pressure within the venous valve. That's skeletal muscle. That's called muscular systole. Okay? All right. So that's the recap on our arteries and veins. Okay, there's a few pulses that I want you to find. And I'm going to go ahead and write them on the board, okay? So I want you to find these pulses. I want you to take a break, and I want you to see if you can find them on yourself. Maybe you get a partner, someone in your family, and see if you can find these pulses. And I want you to just count the beats per minute per each pulse. Maybe pick 2 pulses, write down the beats per minute, and then check them as compared to the other. Is there a difference with the beats per minute? And then lastly, what I would like you to do is go ahead, and this is, if you have a stethoscope, you can do your heart auscultations, okay, and you can hear the heart as compared to the pulse. If you don't have a stethoscope, then just go ahead and use your hand. Put your hand and feel the beats, and then go ahead and feel the beats or the pulse within one of the 2 pulses that you choose out of the ones that I put here on the board. So I'm going to go ahead and write them on the board, and then I want you to go through, take a break, and see if you can find these pulses on yourself or a partner. Better on a partner, okay? Okay, so Pulses of the body. Remember, pulses are arteries. They're relatively shallow, so we can feel the muscles working within those pulses and pushing. All right? And we have five that I want you to find. The carotid pulse up at the neck. Remember, when we're looking for pulses, we don't use our thumb. Why? Because we have an artery in our thumb, a pulse in our thumb. Okay? So we use two fingers. We have the carotid. We have brachial. Okay? We use that a lot on little kids because their necks are relatively small and they have bobble heads, and we really don't want to just push into their neck. Okay? So we have brachial, carotid, and then we have radial. Remember, find your radius. Okay? And then you feel radial pulse, popliteal pulse, back of the knee, and then dorsal pedis pulse. Right on the top aspect of the foot, a little bit more on the lateral side near the talus. Okay? So those are our pulses that I want you to find. So go ahead and take a break and find those pulses, and then count the beats per minute. Okay. I have a link on this module that you can click on this module, and you can really see in detail how to take some of these pulses, and then how we take pulses to figure out the number. Okay. All right. Okay, for any of you that have ever had blood pressure taken, you know that on average we usually get 120/80. So, what does this mean, 120/80? Okay, does anyone have any idea? Where do we usually take blood pressure? All right, there's another video that I'm going to add in here. It's a little bit older, and that really goes in depth. How we take blood pressure. Okay, so I'm not going to go too into detail of how we take blood pressure because I'm going to explain that here in just a minute. I'm going to attach that old video onto this file. But what does this mean? Well, first of all, 120/80. These are numbers, and it's pressure pressed against the arterial walls. Okay, and we record them as millimeters of mercury. That's just the unit of measure that we do. So that is a mmHg, okay? Millimeters of mercury. That's the unit of measurement that we take to measure that pressure. All right? So 120/80, that's normal. That's what we like to be at for human beings, okay? Animals are very different. Giraffes have huge pressure. Elephants have massive amounts of pressure, okay? I want to say giraffe is the highest. Although I don't know, I'm not an animal physiologist. I'm a human being anatomist and physiologist. But so these numbers, first of all, the 120, the top number, this is called 120, or top number, equals ventricular systole, okay? So ventricular systole is when, basically, we have depolarization of the ventricles, all right? So when blood in the ventricles are ejected into the aortic trunk and the pulmonary trunk, we have that BP cuff wrapped around our brachial artery, okay? And this measures the pressure. The BP cuff measures the pressure against the arterial wall. Specifically, when we have ventricle depolarization or ventricle contraction, okay, the QRS wave, this is when we see the spike. This spike, the 120, okay, is when that pressure is at its maximum on the arterial walls during ventricular contraction. So, that's the top number. The lower number, the 80, well, this equals what's called ventricular diastole, okay? And basically, ventricular diastole is when the ventricles relax. So that's the pressure, the blood pressing up against the arterial walls, brachial, during relaxation of the ventricles. You can see why these are important. Because if these are really high, then there's a lot of pressure against the arterial walls. And if these get too high, if we're, let's say, 200/100, nay, my friend, not good. Why? Well, because the pressure can get so high that then the artery tears or cracks. Okay, and then all of a sudden we have an arterial embolism. Okay, or if it occurs in the brain, we have a stroke. All right, so when pressure gets too high, that's what we worry about. We worry about strokes. We worry about aneurysms. We worry about embolisms. Okay, we worry about the arterial wall going past the yield point about how much it can stretch. Okay, and then it tears, or we have damage to it, and then next thing you know, bad news bears. Things are not doing good. Okay, so this is blood pressure. Let's click on over and let's check out how to take blood pressure now.
All right, today we're going to be discussing blood pressure. Blood pressure is assessing the pressure within the circulatory system. There is systolic, which is the top number, and diastolic, which is the bottom number. Systolic assesses the pressure when the heart ejects blood out of the left ventricle, meaning that it's the pressure that is the highest against the artery walls. Diastolic is the pressure when the heart relaxes, specifically the left ventricle relaxes. And that's the pressure on the artery walls when the heart is in a relaxed state. So, first off, we have to look at some of our blood pressure equipment. There's various forms of blood pressure equipment. Here we have, these both are standing sphygmometers, and their pressure gauges are slightly different than what is more traditionally seen, which is the sphygmometer cuff and the gauge right here. With both, or all three, you need a cuff, a sphygmometer dial, and a stethoscope. Okay, the stethoscope will let you hear the beats going through the circulatory system as we pressurize the cuff and depressurize the cuff. Okay, so to perform blood pressure, we take our patient or client, wrap the blood pressure cuff around. You want to make sure that their arm is supinated, and we place the arrow where it says artery, so that it's over the brachial artery. Seal the cuff, and then have it so that the dial is facing forward. Okay, so now once we have the cuff tight and the dial showing, we want to make sure that we get our stethoscope positioned properly. So we're going to have our stethoscope placed directly over the cubital space in the elbow. And we're going to leave that right there. Now, when we pressurize, we want to pressurize to approximately 200 millimeters of mercury. Okay? And we slowly release the pressure. And I have my stethoscope on. And what I'm listening for is for the initial beats to begin. You can see the ticker starting to tick right there. The beats are coming in, and then the beats fade away. When the initial beats occur, around 120 for her, that's systolic blood pressure. That's the pressure that is the highest against the arterial walls during ventricular contraction. When the beats go away, that is diastole, when the heart relaxes and when pressure is against the arterial walls during the heart relaxation phase. So systolic over diastolic. In her case, it was 120 over 80 approximately, which is average. So she's got good blood pressure. That's blood pressure.